Hi everyone. This is the final video related to our Skeletal Muscle Physiology Lab that we're doing in class this week. This one will walk you through Activity 5, which looks at the phenomenon of fatigue, muscle fatigue specifically. Muscle fatigue could have a lot of causes, and actually the true cause is a little poorly understood. There's still a lot of research out there on it. But fatigue is defined in a few different ways, either the inability to generate force in a muscle because of repeated contractions, or a reduction in force over time with repeated contractions. And we saw this in the previous activity, Activity 4. Again, if you haven't watched that walkthrough and done that activity, go back and do that now. But we noticed that we reached a peak even when we stimulated that muscle at almost 150 stimuli per second. We noticed that there was a peak, and then very quickly it started to gradually decrease over time, the force that is. If we were to let that go for some period of time, even a few seconds, we would notice that that force would get fairly close to zero over time. And there are a few theories as to why that happens. One is related to lactic acid. Our ability to clear lactic acid from our muscles does have an effect on performance. But the extent to which lactic acid actually affects muscle force, the actual ability to generate force in a muscle, isn't all that well understood. The other two substances, which there is a little more research to support their role in this process of fatigue, are ADP, and ADP is adenosine diphosphate, which you get when you break down a molecule of ATP and release its energy. And the other byproduct of that process is PI, which is short for inorganic phosphate. So ATP consists of 3 phosphate groups, that's why it gets its name. And when you break the bond between the last 2 phosphate groups and remove a phosphate group, that is how we release the energy in ATP. What we're left with is ADP, which can be regenerated into ATP. Creatine phosphate is actually a really great resource for regenerating ATP. And inorganic phosphate. In fact, most of the research that I found, I just did a quick search about the causes of muscle fatigue, and PI, inorganic phosphate, came up in a lot of those studies. So it's thought that our body's ability to get rid of inorganic phosphate, either by just getting it out of cells or causing it to bind with ADP to form ATP, can help us to reduce fatigue. And so we might see that giving our muscles a rest in between will help us to generate more force in a muscle because we get rid of that inorganic phosphate that may be interfering with that process. So we're going to go right to the experiment here. And this one's a little wonky, so it might give me a couple error messages that I'll need to clear. But we've set the voltage to 8.5 volts, which we agreed was that maximum stimulus. And we've set the stimuli per second to 120. So this is almost where we achieve that maximum tetanic tension, that highest force that we can generate. So we're really hitting this muscle hard. And so we're going to go ahead and do this. And we're going to see how long it takes to get to zero. And here we have time in seconds. So it's a much longer time period that we're working with here. So let's see what happens. So we generate that impulse, and we stop it right there. So we notice that we were able to sustain that maximal force for 0.08 seconds, which doesn't seem like that long, but that can be enough to, for instance, bring a barbell over your head. And so that sometimes is all we need, that very quick, maximal production of force. And so now we see that, and what we're going to see now is that, and so this question is asking us if the cause of this fatigue is the accumulation of acid, of ADP and phosphate, or of getting calcium out of the cell. In reality, probably it's a combination of all three. And so what we're going to look at here, let's just assume that the length of the rest period will not substantially impact the length of time for sustained muscle tension, that it doesn't really matter. And let's see if we're right. So what we're going to do here is we are going to select multiple stimuli. We're going to stop it when it gets to 0. We're going to wait 10 seconds, and then we're going to turn it back on. That will represent a rest period. Then we're going to do the same thing, except on the second interval, we're going to wait 20 seconds, and we're going to see if giving it a longer rest will make any sort of difference. So here we go. I'm going to scroll down, and it's probably going to yell at me a couple times here, but I'm going to try to stay on top of it. So I generate the impulse. It's quick. And now, since the muscle's rested for 10 seconds, I'm going to select multiple stimuli to try to generate another nerve impulse. And we're going to see as we go what the maximum force is going to be. We want to try to get it all the way up to this previous level, which was about 5.86. So, here we go. It looks like we got just about there. And so we're going to stop it. We're going to let it go for about 20 seconds. I almost had it that time. And now we're going to see if we can get up there again. So now we've given it twice the rest. And it looks like we've gotten just about there. It's hard to tell until we collect the data, but let's see what we have. So it looks like, again, we have been able to generate, and let's look at the chart here. So if we had a rest period of 12 seconds here, I went a little over, then we still generated the same amount of active force as we did when we started. We couldn't maintain that force for as long, though. We were only able to sustain it for about a quarter of the time. So certainly our ability to hold that maximal force decreases over time. But we're still able to generate that same active force because we've given the muscle an opportunity to rest. And we notice that with an increased rest period, here we have a rest period of 22 seconds, we have an even longer period where we were able to sustain a maximal force. 0.08 rather, was the starting point where we were able to just start from scratch. We're fully rested. Here we got to 0.06, so we got to about 3/4 of the amount of time that we sustained that force. That's actually pretty significant. That suggests that with maybe 10 more seconds of rest, we might be able to generate that maximal force once again and sustain it for the same amount of time. Because we have time to get rid of lactic acid, we have time to get rid of ADP and inorganic phosphate, and we have time for perhaps enough calcium to come into the cell. So hopefully this helped you out a little bit, and I hope that you enjoyed this lab.